Hey, Rachel. Hey, Rachel. Hey, Rachel. Hey, you guys. My house looks red. I have kids and laundry is over there on the sofa. So, and I just got through teaching a whole bunch of math. You see, to the I'm having fun. Okay, we did chapter 8.
Understood.
Yeah, we did chapter 8 on. We did chapter 8 on Tuesday. I may have to re-record it because it sounded like garbage to me. But what it was,
All right.
the USB port that's on the top of my computer was a little wonky, and the kids messed with it for some odd reason.
Okay.
Loose. So I put it in the back in the day. They everybody said it was much better. So, but we're gonna do chapter 9 today, and when I go over chapter 9, I'm gonna pull it up. I'm gonna probably just have the PowerPoint on screen and turn my camera off.
Okay.
And that's because it just saved a little bit more space, and then you can watch it at a different speed.
Okay. That sounds good.
Okay, who's that? That's AJ. Hey, AJ. He's so funny. I'm gonna laugh at you, AJ. That was funny. How are you doing, by the way? Did you hear me? How are you?
You asking me?
Yes. I was asking
I'm pretty good. I got a head cold, so I'm feeling kind of crappy. But other than that, I'm doing pretty well.
Okay, I understand. If it can just rain so the pollen can go away, I'll be doing much better too.
That would be nice.
Yeah, that's my thing. The pollen is just for me. Okay, we're gonna get started. Let me pull everything up. Okay, Rachel, I'm gonna dim my screen. I'm just like it out. Okay, so if you want to, you can too. Hey Katie, I see you. All right, let me pull up. We're just gonna do chapter 9 today. I'm like, okay, the kids are outside playing. I tried to move them around. They was doing World War III the other day, so hopefully them being outside and running would kind of help mitigate sound a little bit better instead of them being upstairs, because their room is right above my area, and so they were really loud on Tuesday. So we're gonna try to keep them outside and keep them occupied so they don't disturb me too much. All right, chapter number 9. All right, you guys, we're going to start with chapter 9. And chapter 9 really talks about membrane transport. And with that, what we're going to be looking at is basically um, calculating membrane potentials. So a little bit of math in this. Describe ion movements of an action potential. We're going to look at thermodynamics when it comes to these membranes. And then what is the difference between active and passive transport? Now, what about ions and what are ions? And so we know that ions basically, in simplest terms, when it comes to chemistry, is their transfer of electrons. So ions do the same thing when it comes to membrane transport. They transfer, trans in front of it, meaning that. So with membrane transport, we're going to deal with a part that deals with a little bit of signaling as well. Now, what they say is all animal cells, including neurons, maintain intracellular ion concentrations that differ from those outside the cell. Now, if we look at this, and one of the things we look at, and in this figure here, what they're showing is they're showing sodium ions and potassium ions. And these are ions that are in an animal cell. And then you got extracellular sodium ion concentration, and this is about 150 millimolar, is much greater than intracellular concentration, and that's about 12 millimolar, which makes the extracellular K concentration or potassium concentration much less. Now, if the plasma membrane was completely permeable, the sodium ions will flow into the cell down its concentration gradient. And then that's the blue arrow that you see right there on the left. And then the potassium will flow out of the cell, and then the potassium is the orange. Now, so even when you have this, you will have a small percentage of the ions, and they will leak out of the cell. Now, when they do that and leak out of the cell, this generates what we call a little bit of voltage across the cell membrane. So what happens is it has a charge imbalance, and then it generates a voltage. And when it generates that voltage, this is called membrane potential. And so that's what you see right there. And in your book, on page 236, they got some calculations. And what I'm going to do on Tuesday before we have our exam, when we go over chapter 10, I'm going to go over some of this math because I know we need to take a look at it. But in your book, they have some equations that is listed there. And so they have the gas constant R, and that one, and you know, there's a lot of gas constants that are out there. And so you got the change in phi, it should be small phi, is equal to RT over ZF, and then they do the natural log of the ion initially over the ion that's coming out. So they got the gas constant, the temperature is always in Kelvin. Z is your net charge, and you will remember that doing the net charge from General Chem 1 and 2. And then the net charge, and then F is the Fahrenheit constant, and that's the charge per mole, and that's in coulombs, and that's listed there. Now, phi is expressed in units of volts or millivolts. if you got something that's monovalent instead of covalent, but monovalent, Z is always 1 at 20 degrees. And so there are some sample calculations there, and I'll go over those. But just to get to some other things that are in the chapter, and here go the equation right here. So the voltage across the membrane. And so this caused by ion transport. And so this is the equation right here. Next charge, gas constant, Faraday constant. So make sure that you know this because you will see this again and everything. So this is the equation. And like I said, that is changed in 5. Now, what do we do with that? Now, when ions move, they do alter the membrane potential. And so you got 3 parts of that. So on here, you see action potential, you see sodium ion permeability, and then you see potassium permeability here. So in action potential, what happens is you got your inward movement of sodium ions that makes the membrane potential more positive, increasing it from the resting values of around -70 millivolts to as much as a positive 50 millivolts. Now this reverse of membrane potential, or they also call this depolarization, this is called the action potential. Now, the action potential also stimulates the opening of additional sodium ion channels further along the axon. And what they're saying is that's the elongated part of the cell. And when it does that, you get another round of depolarization and repolarization. So you get the transport of basically things going in and out of the cell via the membrane. And so what this chart actually tell you, the neuron membrane undergoes depolarization as those sodium ion channels are open. And so that is shown in the green. That's what they pointed to there. And then repolarizes as your potassium ion are open. And that's in the red. And following the action potential, the membrane may be hyperpolarized. But, and that means just say that it goes a little faster, polarization happens fast, but it returns to normal within a few milliseconds. So these reactions, this transport, it does not take long at all for this to happen. It happens in the result of just milliseconds or even nanoseconds when it comes to that. Now, why do we look at this and why do we care about this? One of the things that we look at is, let's just say we have a nerve impulse and what we look at. So here, they say the stimulus is assumed to originate where the axon leaves the neuronal cell body. The stimulus open up those sodium ion channels into the membrane. And then when you got all of them coming in, that causes the depolarization. Now, when it causes the depolarization, that triggers the opening of the voltage sensitive potassium channel. So the efflux of that restores the resting potential. So that's what you see in this particular one right here in this figure. Now, what else that goes on here? You got some that are active transporters, and then you got some that are passive transporters. Now, the neuronal sodium and potassium channels are considered passive transporters, and that's because they provide a means for ions to move more down a concentration gradient. And with that concentration gradient, it's more thermodynamically favorable in this situation. And so it's a thermodynamic favorable event. Now, what else is going on? So when that initial depolarization also triggers the opening of that, they move further along. So they're moving further down. And then the action potential moves down the length of the axon as a wave of depolarization and repolarization. This travels in only one direction because the previously open channels remain closed. And so that is what's going on right here. Now, what you also see, this myelination of an axon, and then right here they show that's a scanning electron micrograph of that right there, and that's the electron scanning or transmission. No, that's transmission. Micrograph of a myelinated axon, and so that's what you see here. And it's just a cross-section diagram showing how the accessory cell pulls around the axon. So you see that, and that pulls around it, and that multiple layers of this plasma membrane coat the axon. And then the electron micrograph shows this, and the sheet may be 10 to 15 layers thick. And so on there, on this one, if you look closely at the one over there in the corner, if you count them, that is roughly about 10, and then that's in the lower right corner, and then in the upper left corner, that one is more so around 15, but the one that's in the middle, I don't know if that, it may be right at 10, it might be more so 9 layers, but that's what you're seeing there. Now, what they're going to do is in the chapter, and like I said, it's a lot of math that we're going to have to go over. On page 239, they are showing calculations, and one of the things that I wanted to point out on page 237, they talk about Gibbs free energy. And with Gibbs free energy, you got RT, the natural law of a substance X, N over it going out, the substance going out, the concentration of it. So you guys are going to be calculating Gibbs free energy of that particular one. So they have a few calculations on that in your book on 239, and you should have some also in your homework as well. So make sure you take a look at those, and I'll do some calculations on that as well. Now, one of the things that I want to get to before we get into passive transport is talk about, when we were talking about the potassium and the sodium ions, I wanted to contrast something, excuse me, with you. And basically says that the proteins that initially establishes and maintains the cell's sodium and potassium ion gradients is an active transporter that needs the energy of ATP hydrolysis reaction to move the ions against their concentration gradients. So with passive transporters, you also need your active transporters. So that's one thing I want you to keep in mind. Not only with active, with passive, you also got to have active transporters. And so you need to just keep that in mind. So our next section, what we're going to talk about is passive transport. Now, so when we talk about that, transporters of one kind or another have been described for virtually every substance that cannot be easily diffused through a bilayer on its own. So some transporters can go straight forward through the membrane. Some of them they're going to function much just like enzymes. So we're going to start with passive transport systems. So we're going to start with porine. And so porine has, here you see the stick model and you see the ribbon model that is there. And now, porins are located on the outer membranes of bacteria, mitochondria, and chloroplasts. Some bacteria and organelles descend from them have a second outer membrane in addition to the membrane that encloses the cytosol. Now, all known porins are called trimers, in which each subunit forms a 16 or 18 stranded membrane spanning the beta barrel. So you guys see that there. Now, a beta barrel of this size has a water-filled core with hydrophilic side chain. So here, that's what you see, the E. coli of porin right there. And each subunit of the trimeric protein forms a transmembrane B barrel that permits passage of ions or small molecules. Now, with that, now, one loop make the porins specific for small, and these are catatonic solids. So anything that is catatonic, so sodium, potassium, those type of things. So that's what you want to look for. Now, in the 16 strain one, long loops connect to the beta strains. Here, one of the loops of each monomer unit folds down the beta barrel and constricts its diameter to about 7 angstroms at one point. So what it does is when it constricts it, that means only certain substances can pass through. And that the ones that can pass through, they can't be larger than 600 D. So the loop bears several carboxylic side chains, which makes porine weakly selected for its catatonic substance. Okay, so with that, other porins exhibit a greater degree of solute selectability or selectivity, depending on the geometry of the barrel. So the geometry, the size of it, that allows for transport. So you have to look at that and look at size. So that is important. Now, when it comes to these ion channels, they are very selective. Now, this is the structure of your potassium channel here. Now, from Lividans. So 4 subunits are shown in different colors. So you see that there. Each subunit got mostly an inner helix that form part of the central core and an outer helix that contacts the membranes interior. And so that's what you see there, and you see all of that right there in the middle. Now, when I was saying about that, the ion channels are highly selective. When you got eukaryotic and prokaryotic cells, they are more complicated proteins than the porins. Many are monomers of identical subunits. They have alpha helix membrane spanning segment. And so here, what you see is this is potassium ion channel, and this is a selectivity filter here. And then this model shows a portion of the pore looking from the extracellular space into the selectivity filter. The pore is lined by a backbone of carbonyl groups with a geometry suitable for coordinating a potassium ion. And the potassium ion is in that purple sphere. Now, purple sphere in shape, if you remember from earlier chemistry, S, P, D, and F, and that's S because of the potassium, and that's a purple sphere. Now, a rigid protein network that includes tyrosine residues prevents the pore from um, contracting to accommodate the smaller sodium ion. So the atoms are color coded. So here you got carbon in green, you got nitrogen in blue, and you got oxygen in red. And so that's what you see there. Now, with that, I think what is, all right, so just one thing that I want to go back to this right here. The high selectivity for potassium ion reflects the geometry of the selectivity filter. And then they say it's an arrangement of protein groups that define the extracellular mouth of the pore. So I want you to keep that in mind. Now, when you talk about operation of the voltage-gated potassium ion channel, this is what you see here. Now, this is a S4, S5 linker helix. And so the S4, S5 healer linkage is shown in red on both of those. And it pushes down on the X6 helix. It's pinching off the intracellular end of the pore. When depolarization occurs, the linker helix swings upward and the X6 helix bends and opening up the pore. So that's what happens in that case. Now you got channels here, and you got these conformations. Now, gated channels undergo these conformations. Now, what they say here that if sodium, if potassium ions or sodium ion channels were already open, the nerve cells would not experience action potentials. So the intracellular and extracellular concentration of ions will quickly regain equilibrium. When they do that quickly, they'll kill the cell. Consequently, those channels and many other are gated. Now, they have to be gated because they got to open and close when they need to. Now, some channels respond to different things. One of the things they respond to is pH, or the binding to a specific ligand, such as the calcium ion or small molecule. Some channels proteins open when phosphorylated or have a phosphate group covalently attached. So sometimes the phosphate groups can open them up. So in this case right here, and these are just closed and open ones. So in the bacterial proteins here, you got MSCS and MSCL. These are sets of alpha helixes surrounding the pore in the rest of the proteins. Those are not shown, but the helices on the side past each other, opening and closing the pore, much like the iris in the eye. Okay, so hydrophobic residues that block the pores in closed state are shown in magenta. So that's what you see there. Now, what about if you have aquaporins? So aquaporins are specific, aqua meaning water. And so water molecules were assumed across membranes by simple diffusion or by osmosis. But because water is present in a large amount in biological systems, think about it, our bodies are about, 69% water. 70% somewhere up in there. This premise seems reasonable. However, certain cells, such as the kidney, can sustain unexpected rapid rates of water transport, which suggested existed of a previous unrecognized pore for water. So, when it comes to this protein, this was discovered in 1992 by Peter Avery, which coins the term aquaporin. Now, aquaporins are widely distributed in nature. Plants have a lot of them. They say 50 different ones. There are 13 mammal-based ones, and they are expressed at high levels in tissue where fluid transport is important, including in the kidneys. You also have them in salivary glands, lacrimal glands, and those are the ones that produce tears in your eyes. Now, those are most of those are extremely specific for water molecules and do not permit transmembrane passage of other small polar molecules, such as glycerol or urea. And so, this is what you see here. So at the top, the ribbon model is viewed from within the membrane. And then B is the stick model of it, viewed from one end. So in the intact aquaporin, 4 of those subunits associated via hydrogen bonding, because you got water in there, so you got hydrogen bonding going on between the helices and through interactions among the loops outside the membrane. Now, this is a view of an aquaporin pore right here. You got hydrophobic residues are colored yellow, and then the 2 asphodel residues are red, and you see those in there, ASN 192. Now, so unlike the potassium channels, whose pores lies in the center of 4 protein subunits. Now, each aquaporin, it contains a pore, so each subunit contains a pore. Now, they vary. So one of them varies around 3 angstrom in diameter, and then the diameter of water molecule is around 2.8 angstroms. And the dimensions of the pore, it clearly restricts anything else larger than that. Only that water molecule and something smaller than the water molecule can get through. And so that's what you see here. Now, when you got transport proteins, they alternate between conformations. So let's talk about this one. So you got an operation of the red blood cell glucose transporter right here. So when you see this, you got your glucose, and that glucose is hexagon. It binds to a site at the transporter face that faces the cell exterior. So it goes in and it binds there. So that's one thing that it does. And then the glucose binding triggers a conformational change. So in this case, what it does is, at the top, when the glucose went in, it looks like it's open. And then what it does is it kind of loops around it and closes and then opens at the bottom. So what it does is it accepts it in and then it closes it at the top. So it exposes the glucose binding site to the cell interior. And then it looks like, the glucose just flows out. Now, the glucose dissociates from the transporter, and that's what it does there. And then once it does that, the transporter goes back to its original conformation. So think about this. That just one glucose transporter, that's what they're going to be doing if you've got a lot of glucose molecules. It just opened up, closes, transported. And this does this within milliseconds. It doesn't take a long time to do this. So that's what it does. It open up, take the glucose, move it, and keep going. So it just accepts it in, push it out. Accept it in, push it out. That's basically what it's doing right here. Now, you have different types of membrane transport systems. You got uniport, you got symport, and you got interport. So the uniport or the uniporter is such as the glucose transporter, move a single substance at a time. You got a symporter, that's the same one, the symport, transfer two different substances across the membrane, and then your antiporter moves different substances in opposite directions across the membrane. So you got the uniporter that, like the glucose one, they just take one in, drop it out. Take one in, drops it out. You got your symporter that takes two in, drops them out. Take two in, drop them out. And then your antiport or antiporter, one comes in, one goes out, and they're going in opposite directions. So they're not going in the same direction. Uniport and symport, they go in the same direction pretty much. And then your antiport, they go in opposite directions. And so that's what you see here. Now, let's talk a little bit about active transport. So we talked about passive transport. Now let's talk about active transport and what's going on with active transport. Now let's take a look at this figure right here. This figure is the reaction cycle of sodium and potassium ATPase. So what we're going to look at that is, as the name ATPase implies, this is ATP hydrolysis in its source of free energy. Other ATP requiring transport proteins, they pump a variety of substances against their concentration gradients. So this is what you see here. So in step one, you got three intercellular sodium ions. They're going to bind. So they go through ATP, and then the ATP binds, and then you see that. And so the phosphoryl group is transferred from ATP to an ASP side chain of the pump. The ADP then is released. We talked about that earlier in the previous chapter. In the protein conformation chain, exposing the sodium binding site to the cell exterior. Now, with that, the sodium ions, they dissociate, they separate out. And then now when they do that, the extracellular potassium ions bind. And then what happened is you got your aspartyl phosphate group is hydrolyzed, and then your PI release. So you got a protein is released. So your protein conformation changes, exposing the potassium ion binding sites to the cell interior. Then here in this case, that's where your potassium ions dissociate, and then the cycle goes over and over and over again. And so that is what happens. So when you look at this, this is energy based on energy wise. This is favorable. This is what we want to happen. Now, let's look at this structure. Now, this is the structure of the sodium potassium ATPase. This subunit is green, in the beta and gamma subunits are blue. 3 sodium ions are there. They are in orange. So they're right there in the middle of the membrane. They occupy the ion binding site, and an ATP analog is in yellow, marks the sites of phosphorylation. And that's down there at the bottom in this cytosol. And that's what you see. Now, this is known as P-type ATPase. P stands for phosphorylation. Other types of ATP-dependent pumps are the V-types, which operate in plant vacuoles and other organelles, and the F-type, which is usually operated in the reverse to synthesize ATP in mitochondria. So that's what you see there. Now, what you see here is mouse flea glycoprotein. Now, what you see here, now some cells or all cells have the ability to protect themselves from toxic substances. Now, that insert the cells in the lipid bilayer and alter the membrane structure of function. Now, this defense depends on the action membrane proteins known as ABC transporters. Now, ABC refers to an ATP binding cassette, a common structure motif in these proteins. Now, when it comes to this, you got a lot of antibiotics and other drugs that are lipid soluble, and therefore these substrates are used for the same transport. Now, drug resistant in cancer chemotherapy and antibody resistant in bacteria have been linked to expression or overexpression of ABC transporters. In humans, this transporter is known as P-glycoprotein in the multi-drug resistant transporter. And so that is what you have. Now, this is the structure of it. It's built from a single polypeptide chain. Now, this is the ribbon model of it. Note that the internal cavity is open to both cytoplasm and the inner leaflet of the membrane. So that's what you see here. Now, one thing that we have to keep in mind, ABC transporters function much like other transport proteins and ATPase pumps. The ATP-dependent conformational changes in cytoplasmic portions of the protein are coupled to conformational changes in the membrane-embedded portion of the protein. So this is what's going on. We have all these different types of conformations, and we talked about conformations and everything in organic chemistry. And you have this actually going on within the cell. So you got these that is actually doing that. Now, what you see here is you see glucose transport that is in the intestinal cells. So what you see here, the sodium K ATPase establish a concentration gradient here in which the sodium ion that's going out is less than the sodium ions going in. Sodium ions move into the cell down their concentration gradient along with glucose molecules via a SIM-port protein that transfers sodium and glucose simultaneously. So the glucose and the salt is going down at the same time. So the salt and the sodium ion and basically the sugar. So let's just say you got salt and sugar in your body. And so sometimes when they're transporting it, just think about your kidneys, and a lot of times with people when they eat a lot of salt, or especially a lot of sodium, and your body cannot handle all of that sodium. And after a while, that's where you see swelling or edema that happens. And I know with me, personally, I can't eat a lot of salt, because if I do, I know that my legs and stuff will swell, and I see swelling. And so when you see that, you know that's edema. And I know especially for my family, that's critically important, because my dad died of a heart attack, And my sister just had one a few weeks ago, and she's getting ready to have open heart surgery. And so I know in my family we have that history of heart conditions and stuff like that. So I know one thing, especially for me, I have to watch my sodium intake because I know my body can't handle that. And I know even with some allergies and food allergies that I have, especially some seafood that I can't eat, is because of the sodium that is in there. Sometimes I do good, sometimes I don't do as well. So that's one indication, and so you can look at that. Now, going back to this, glucose thereby becomes more concentrated inside the cell. When it exits down the ion, its concentration gradient via passive uniport blood transporter. Energetically favorable movements are indicated by green arrows, and the energy requiring movements are indicated by red. So you see those at the bottom, and you see the glucose coming in. But what it's trying to do is go and goes through that, and it goes into the blood. So that's why people have to prick themselves to check um, their um, their glucose levels when it comes to things like diabetes. Now, that's one thing that we're looking at. Now, one other thing that we're going to look at and talk about really in this chapter is membrane fusion. Now, what happened to the membrane and why do they fuse? Now, when you talk about um, membrane fusion, what we're looking at is the final steps in transmission of a signal from one neuron to the next, or gland or muscle cell, that culminate to the release of substance known as those neurotransmitters from end of the axon. Now, common neurotransmitters include amino acids and compounds derived from them. So when you have the synapse linking motor neuron in its target muscle cell, the neurotransmitter right here, and that's what you see is acetylcholine. And acetylcholine is a common neurotransmitter. Now, acetylcholine is stored in a membrane-bound compartment called synaptic vesicles, and they're about 40 nanometers in diameter. Now, when this action potential reaches the axon terminus, the voltage-gated calcium ion channels open up, excuse me, and allow the influx of those calcium ions. Now, the increase in those local intracellular calcium ion concentration, they are less than 1 micromolar to as much as 100 molar, and it triggers exocytosis. So that's what other vesicles. And so basically, you see this here. Acetylcholine is degraded at the neurosynapse by its named protein right there. So that's what happens. And you get your acetate and your choline right there. Now, so you see this right here. So they show this via the reaction. Now, what happens when this happened at the nerve and muscle cells? Now, with this, what happens is you got an action potential reaches the axon terminus. So that's what these channels here, they start to open. So that's what you see in there. So you got your muscle cell, you got your receptors, they start to open. Now, what happens is that triggers the fusion of the synaptic vesicles with the plasma membrane. So when it does that, that's what the acetylcholine is released into the synaptic cleft. So right here, you see at the top, basically they are moving over, and then they're going to open up, and then they're going to be released. And then the acetylcholine binding to the receptors on the surface of the muscle leads to muscle contraction. Now, that signal is short-lived because the acetylcholine remains in the synaptic cleft. It's rapidly degrading. So that's what's going on with that. And then you have muscle contraction. Now, looking at this, you have snares that link vesicle and plasma membranes. So this is a structure or a 4-helix bundle of the snare complex. There are 3 proteins. One includes 2 helixes are in different colors. Portions of the snares that do not form the helix bundle will cleave out before X-ray crystallography. And so that is what you see here. Now, just to talk about snare, they're integral membrane proteins. The name comes from the term soluble in ethyl melamine sensitive factor attachment protein receptor. So 2 snares from the plasma membrane and 1 from the synaptic vesicle form this complex that include, and it's about 120 angstrom long, full core structure. Got those 4 helixes in there. Now, the 4 helixes have about 70 residues. They line up in parallel fashion, so that's what you see there. Unlike other core proteins, such as keratin, this 4 helix bundles is not perfectly geometric structure, but it varies irregularly due to the diameter of it. Now, with that, now, when you have snares and everything, their membrane fusion is spontaneous. Now, so when they have it, the formation of the complex of the snares from the vesicle and the plasma membranes bring the membrane close together so that they can fuse. And so, because of that, the formation of the snares complex is thermodynamically favorable. Membrane fusion is also spontaneous in this case. Now, when it comes to different types of experiments, however, when you got pure lipid vesicles, they demonstrate that snares are not essential for membrane fusion to occur in vitro, but the rate of fusion does depend on the membrane's lipid composition. So here, what you see is basically the schematic of this. And what they did was, to keep it simple, they kept the vesicles in the plasma membrane. They're depicted as bilayers. And so that's what they did here. I'm trying to see if there's anything else. Okay. And so you have endocytosis. And so with endocytosis, you got pinocytosis capture right here, the extracellular materials, non-specifically in the receptor mediated endocytosis. A molecule binds to a specific receptor, which is then internalized with its bound ligand. Co-proteins just beneath the membranes interact with the receptors and help mediate the membrane shape required to pinch off a portion of the cell membrane to form a vesicle. So this is what you see here. And then your Catherine assembly here. These this is 3 subunits. They're shown in a model that was taken from an electron micrograph. And then you got multiple subunits assembled to form a lattice that surrounds and defines the shape of the vesicle itself. Now, just a few things, because this is the end pretty much of this chapter. And so just to wrap up things, the transmembrane movements of the ions generated changes in the membrane potential during transport signaling. So that's one thing to keep in mind. The free energy changes for the transmembrane movement of a substance depends on the concentration on each side of the membrane. And if the depends on the concentration on each side of the membrane, and if the substance is charged, the membrane on the membrane potential. Now, you got passive transports and you got active transport. Now, passive transport proteins such as porins, and remember we got porins, we got aquaporins, they allow the transmembrane movement of substances according to their concentration gradient. Aquaporins, aqua meaning water, they transport water molecules. Now, ion channels, they have a selectivity filter that allow passage of one type of ion. Gated channels open and close in response to some other event. Membrane proteins such as passive glucose transporter undergo conformational changes, so you will see those that alternatively expose to ligand binding sites on each side of the membrane. Now, you also got what we call active transport. Now, active transporters such as sodium K-adipase and ABC transporters require free energy of ATP to drive the transmembrane movement of those substances against their concentration gradients. Then you got your secondary active transporters allow favorable movement of one substance to drive the unfavorable transport to another substance. Now, last thing that we just went over are the snare proteins. They're in vesicles and they target membranes to form a four helix structure that brings the fusion of the membrane close together. In endocytosis, a vesicle buds off from an existing membrane to form an intracellular vesicle. All of you guys' work should be up. I know I had to go through yesterday and re-upload chapter 6. It was doing something kind of funky, but I got it re-uploaded. You should already have your notes from chapter 6 through 10. As far as your final exam and other exams, Dr. Craig sent me a list of topics that should be covered on your exam. So now, if we do not go back to school, I don't know what's going to be going on, but if we don't go back to school, you will have your exams online. How your exams are going to look online. It's just like you've had the quizzes before. You're going to have quizzes. It's basically you're going to have multiple choice, about 40 to 50 of those. And then what I'm also going to have you do for the math part of the exam, because this particular exam is going to have math on it. I'm going to upload the sheet in PDF form or whatever, or Word form. You need to write that out and re-upload it and scan it back to me so that I can print it out and grade that. So you're going to have one part that is multiple choice that's going to be totally online, and you're going to do that when you take your exam, and then you're going to have the rest of the day to do the other part of the exam. So when the exam opens, you're going to be timed just like you do it if you're in class. So you have 80 minutes to do your exam. And then the other part of it, you just have to turn it in by midnight that night and probably what I'm going to do is when you take your test, what you're going to do is basically it's just going to be open for that day. That's it. So I'll probably open it up the night before. You basically going to have 24 hours to take the test. Is basically what I'm going to give you. So I open it up that morning. It'll open up. You have 24 hours to take the exam. That's basically what you're going to have. The written part, you're just going to have to scan that. It's in that back to me. Like I said, Dr. Craig gave me a list of topics that should be discussed. I'm going to look at that. I will give you guys hints on those topics, and so that you can see what it is that you need to study for your final. And what I'll have to do is make sure those topics are covered on your final. As far as the amount of questions and everything on your final, if I have to make the final now, I will put more questions on the final. So instead of 40 questions on the final, you may have 60 to 70 questions on the final. That just give you more questions on the final to have. And I'll make sure that I'll do some type of study guide or something to give you some guidance on what would be on there. So don't worry about that. I'm going to go. If you guys got any type of questions or anything, let me know. You can certainly put it in the chat. I'll look at that. Send me an email if you got any questions, homework or anything like that. I'm trying my best to work out a way to get do office hours, and like I said, my kids' schedules are thrown off a little bit because they're at home all the time. So I'm trying to kind of work that out a little bit for them, so that I can get to you guys, so that if you got questions or anything. But I'll make sure that I get some quizzes and some different things up for you guys, so you guys can be ready for your next test. On Tuesday, what we're going to do is go over chapter 10. I want to finish up chapter 10. And then what we'll do Thursday is review Thursday. And then I'll put the test for the next Tuesday. So that should be on the 6th or 7th. So that's what we want to do. So I want to have a day that we actually go, and like I said, I got a board here, I got a setup here, so that we can go over some of the math, so that I can make sure you guys understand and know the math. So there's going to be a part of that that's going to be on the test. So what I want to do is go ahead and get finished, and with chapter 10, go over the math from chapter 7 and chapter 9, and so that you can be ready for that part on your test. Like I said, I had planned the test, Rachel, for the 2nd, but I'm thinking I'm just going to put it back for that Tuesday on the 6th. So that's what I'm going to do. So it should be the week of the 6th. So the 7th, let me pull up my calendar real quick. Yes, I had planned it for the 2nd, but what I'm going to do is I'm going to review on the 2nd, and then that'll give you guys time to study and do it on the 7th. So that's going to be April 2nd. Okay. No, you're fine. It's okay. Any other questions that you guys may have? Chapter 8. If you review it, it sounded kind of crazy. I'm going to probably re-record chapter 8 because it sounded boggled to me and it didn't sound that great. So I'll probably re-record that so you guys can have that. Feel free to review the videos and everything. Like I said, it takes about 20 minutes depending on how my internet want to act, because I got satellite here, and that it will take to convert over to the MP4 and go over to Dr. Wanda Jones classes videos on YouTube. That's where my videos, that's where I'm putting them at. So Dr. Wanda Jones classes videos. So I am going to put them there so that you can have them. And if you need to go back and review, you can. You also have the PowerPoints, you got the book. Like I said, I'm going to try to get some review material together for you guys so you can be ready for the test. And everything, so you guys can be ready. So like I said, next Tuesday we'll finish up chapter 10, and then Thursday we'll just review the math. So that class will be Thursday, just math review on next Thursday, and then we'll have the test on Tuesday. On that Tuesday, I'm just going to come on for a brief, this is the exam, this is what you expect, blah 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 blah, and then I'm going to be done. And then you'll have pretty much the rest of the day to do the exam. So I'll try to make sure that I have it uploaded that Wednesday night, so that it'll be uploaded Wednesday night. And then you'll have all day Thursday to complete the exam. That's all that I have for now. I will talk to you guys later. You guys, I miss meeting with you guys and everything. I wish my kids could be in daycare because they're driving me up the wall. I love them, but they are very, 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 very active right now. So I'm about to go check on them really quickly to make sure they are okay. It is somebody out there with them and everything, but I will talk to you guys later. Like I said, if you got any questions, let me know. And I will see you guys next Tuesday. Have a good one.